Hello, everyone. Welcome again to Life in Space Conference. I'm Nikola Bukowiecka from Polish Astrobiology Society, and I have with me Professor Francine Garrett-Bakelman. Hello.
Hi, thanks for having me.
Yeah, we appreciate your presence here. It's great to have you. So, Professor Francine Garrett-Bakelman, she's an assistant professor of medicine and biochemistry and molecular genetics at the University of Virginia School of Medicine. She's a hematologist and an oncologist studying molecular and cellular biology events in disease establishment and progression and genetic changes in acute myeloid leukemia. And she's also a lead author of the NASA Twin Study original scientific report, and also a contributing author to several follow-up studies utilizing data from the NASA Twin Study since. And she will be presenting some aspects of this work for us today. So we're thrilled to have you again, and the floor is yours, Francine.
All right, wonderful. Can everybody hear me? Everybody hear me okay?
We can hear you. We can see your presentation.
Excellent, excellent. Well, welcome everybody. I know it's been a really interesting meeting. I've attended some sessions here and there and have been very impressed with what people have had to share and everything that you guys are doing. It's really wonderful. And I wanted to thank the meeting organizers for inviting me today to participate in this meeting. As mentioned, I'm an assistant professor at the University of Virginia. I'm a physician scientist, but I had the opportunity when I was a postdoc and during the first couple of years after I moved to the University of Virginia to participate in this very unique study, the NASA Twin Study, and I'm really happy and thankful to having the opportunity to come today and share some of the information from that particular research study that I had the privilege of participating in. And so the goal for the next 45 minutes or so is to discuss and introduce the NASA Twin Study for those who are not familiar with it, and to talk about a couple of the biological mechanisms that we think we identified in that study to be affected by long-term space travel. I always like to start by thanking the people who were part of the project and the subjects of the study. And so I have no financial conflicts to disclose. At the time that I participated in this study, Christopher Mason and Ari Melnick were my PIs as a postdoc, and Christopher Mason was really the PI at Weill Cornell Medicine who was awarded the NASA funding to participate in this research study, and a variety of individuals that I worked with in both Ari and Chris's lab during that time. I'd like to thank the Twins working group that I'll be introducing in the next couple slides, as well as the NASA Twin Study directors, leaders, and staff without whom this research could not have been possible, and of course the study subjects without whom the entire concept could have been realized. And this research that I'll be discussing today was funded by NASA. So the NASA Twin Study Working Group was a set of 10 research labs in the United States who came together to do a collaborative research project on what NASA termed the Twins research study. And the institutions from where the labs were are listed here in the mission logo. And here are images of all 10 PIs that participated in the study. And as mentioned, I worked with Christopher Mason, but the areas of the research project that I participated in really integrated with almost all of the research PIs that were part of the research team. And we were led by 3 individuals from NASA, highlighted here, Craig Kundrat, Graham Scott, and John Charles. And without their leadership, this particular research study would have never come to fruition. So at the essence of the Twin Study is this question. So there are plans to get people to Mars in the not so far future, in the 2030s. And the question is, how will we get people to Mars? And that has a lot to do with engineering and developing the vehicles to get people to Mars. But the question also centers around this, and that is, how will humans actually survive this mission to Mars? This mission takes 2 to 3 years, and it involves clearly a lot of steps to get there. And really, what we know about how space travel impacts human biology is really limited to people who've traveled to the International Space Station and the very few people who've actually traveled to the moon and back. And so to understand how people will do from a physiological and biological perspective in this more long-term space mission really motivated the twin study to be done. And when you think about this type of a journey to Mars, you have to think about a variety of parameters and features that astronauts would be subject to, and they include both environmental considerations and human biology considerations. And so I mentioned that this would be a 2 to 3 year mission. There is an increased risk of radiation exposure, increased risk of injury, exposure, of course, to microgravity and altered gravity conditions, and not to mention the confinement and isolation that individuals would be subject to during this long-term mission. And in terms of human biology, there would be known and unknown physiological challenges to humans and psychological conditions and challenges to humans, and health risks, whether they're mediated by space travel or whether they're mediated by personal risk that people would develop certain diseases even if they were on Earth during that time. And so underneath all of these potential risks and potential for a human on Mars, of course, is our human biology and physiology, which is, as you know, quite complex and involves a variety of systems in which multiple cell types play a function, and in which numerous biochemical and other types of reactions take place. And fundamental to all of those, of course, is the dogma of life, where you have your DNA transcription to RNA and translation to protein, which can be regulated by a variety of mechanisms, including but not limited to epigenetic mechanisms such as the epigenome itself, where there's modifications to the bases in the actual genetic code, to epitranscriptome that can be due to modifications of the RNA molecules themselves, in addition to things like expression of microRNAs or enhancer RNAs that can impact gene transcription. And lastly, but not leastly, the epiproteome, whereby there's a variety of modifications to proteins that associate with the genome and the transcriptome, and their modifications can further influence how genes are translated and how they function in our cells. So at the core of the NASA twin study was the question, how does long-term space travel really impact some of these very fundamental biology processes? So to study this, the subject of the NASA twin study lived on the International Space Station. And I'm sure most of you, if not all of you, are familiar with it, but I just wanted to highlight a couple of things about the International Space Station. So most people who travel to the International Space Station are there for 3 to 6 months at a time. And the size of this facility is about the size of an average 5-bedroom house. They have a very unique lifestyle and perspective, where they live about 270 miles above the Earth, and they orbit 16 times a day. So, in terms of circadian rhythm and exposure to sunlight, this is a very different type of lifestyle compared to what we experience on Earth. They're traveling, of course, at a high speed of about 17,000 miles per hour, per hour. And the other thing that I'll mention, which is important for the interpretation of some of the information that I'll be presenting and discussing today, is that the International Space Station is still within the protection of Earth's magnetosphere. Despite these limitations and this unique environment, on the International Space Station and on other types of missions that have been conducted into lower Earth orbit, there have been numerous models of disease and models of biology that have been studied. And highlighted here is a image from a review article that discusses some of the models that have been studied in this type of environment. And what I'm not going to go through all of these, but what I want to highlight is that humans are a model system to study on the International Space Station. And from our perspective, from the NASA Twin study, investigators, we felt that it is one of the best models to study on the International Space Station to understand how space travel can impact human biology and physiology. It is not still the perfect model to study because very few people go into space and travel. And so it's a very limited representation of human biology that can be studied. Because of that, there's limited diversity of the actual people who can be studied on the International Space Station. There are limited types of experiments that can be conducted due to ethics and the environment on the International Space Station. And there is a lack of appropriate controls that can be studied in this context. And I think this is where the NASA Twin study is unique, is because we really thought that we had the best of all possible controls that we could have for studying human biology during space missions. So the NASA Twin study officially launched into space in 2015. However, there was a period of time of about 2 years in advance of that where there were a lot of preparatory and work towards completing the research investigations planned on the NASA Twin study. And as mentioned, this was truly a collaborative effort between 10 different research labs. And we focused on the study of 2 specific individuals. And so the Kelly brothers, who are represented here, they were both Navy test pilots. They were both astronauts. However, by the time the NASA Twin study was completed, Scott had about 10 times more time flown in space or lived in space compared to his brother. The important thing is that they were identical twins. And so the hypothesis was that anything that we would observe to be different between them could be attributed to a long duration mission in space. And the plan for the NASA Twin study was for Scott to live on the International Space Station for approximately 1 year. And during the NASA Twin study, what we compared is data from each of the brothers during the entire length of the study, which was about 2 years, including pre-flight, in-flight, and post-flight time points. And then within Scott, the in-space specimen collections compared to the pre-flight and post-flight, as well as comparing all of the data from Scott to his brother Mark. And so that was kind of the overview of the study design. And each of the labs that participated in this study really focused on different aspects of human molecular biology. And I do not have time today to review all of the findings from even the original Twin study report, but I will focus in on a couple of interesting findings that were identified by the labs highlighted here, and how we tried to integrate those results to come up with hypotheses about the influence of a longer-term mission, meaning a one-year mission in space, on human biology and physiology. I think this study was unique, and one of the most unique aspects of it is that, to our knowledge, it was the first multi-omics approach to assessing human health and responses to long-duration spaceflight. So how did we actually do this study? And so I was involved in the collection of specimens, processing, and then subsequent data generation and analyses. But my main involvement involved the use and production of data from blood specimens. But in general, we had blood, stool, and urine specimens collected. And those that were collected pre-flight and post-flight were processed immediately for the downstream assays, but then were also, some of them were frozen away for later processing. And I'll explain how that kind of built into the study design in a couple of slides. And this is what you would typically think about if you were collecting specimens from human subjects on Earth, right? This is typically what you would do. But the challenge for us is that clearly the ability to do fresh processing on the International Space Station was limited. And in order to have equivalent specimens collected on Earth and on an International Space Station, we had to come up with kind of protocols that could be performed in both spaces. And so, as you can imagine, right, if you're trying to do an experiment in the lab, a bench experiment in the lab, you know, these are some of the things that you would typically have around you. And this is a movie taken of Andy Feinberg, who is one of the PIs in the twin study. He flew on the hyperbolic planes and tried to do some of these pipetting and volume transfers while they were in the weightless phase of the flight. And as you can see, it is not quite easy to do many of these things in that situation. And that clearly would create a challenge for performing even very standard types of specimen processing on the International Space Station. So something like this, at least at the time that we did our study, was clearly not a good option. So to our advantage, though, there are freezers on the International Space Station. So we could take blood, stool, and urine specimens and freeze them away so that ultimately those specimens could be returned through Soyuz capsules after landing to the US for sample processing in Houston, Texas. And myself and several of my colleagues that were involved in the study actually were the people who flew to Texas and actually processed a lot of these specimens as they came through. And so what the study really at the end was composed of were pre and post specimens collected from the subjects. Some of them were freshly processed, some of them were frozen, and then same thing happened on the International Space Station. Mainly, most of the specimens were frozen and returned back for processing. But on several occasions, we had opportunities to time specimen collections with Soyuz returns. And so those specimens were never frozen. And we called those the ambient return specimens for direct comparison to the fresh processing that we did on Earth. So through these approaches, we collected various data from a variety of categories of biology and physiology in the pre-flight, in-flight, and post-flight periods. And then we integrated this information together to really come up with some conclusions and possible hypotheses about the effect of spaceflight on human biology and physiology. And like I said, I do not have time to review all the results today, and the original result came out in Science in 2019, but there is a slew of papers that came out in 2020, and I put the website here for those who might be interested in reading some of the follow-up studies. And there have been numerous additional studies since that have either utilized the NASA twin study or that have built upon some of the hypotheses that we proposed in our original study as well. But in the interest of time today, I'm going to focus in on 3 aspects of the study that I thought are really interesting to discuss and review, and I hope that you find them interesting as well. And that has to do with telomere length during spaceflight, immune and inflammatory responses, and then mitochondrial function and stress. And the data that I'll be sharing today comes from these 4 papers that I highlight here. So this is kind of a map of all the specimens that we had collected during the original twin study and that we reported in our original report. And in here and throughout the presentation from now on, anything that's labeled in green is a specimen that came from the Earth twin. Anything labeled in blue or purple is a specimen that came from the flight twin who flew on the International Space Station for a year. We had specimens pre-flight, in-flight, and post-flight. And then we collected them, as I mentioned, from the blood, the urine, fecal specimens. We also had physiological measurements of vasculature and cognition, ocular measurements, and body physiology parameters. But I'm not going to have time to discuss all of these today. And as I mentioned, there were fresh processing specimens, frozen specimens, and then the ambient specimens. And the way that we processed the blood is we collected blood either on Earth or on the International Space Station into CPT tubes. These tubes are useful because they create a closed system with a polyester gel that can actually separate the different components of the blood when you spin them in a centrifuge. And there is a centrifuge on the International Space Station in which these could fit. And so they were processed the same way on Earth and on the International Space Station to separate out the plasma and the cell types. And then some of them were frozen, both on the International Space Station and on Earth. And some of them were processed freshly to isolate plasma for antibody and cytokine detection, and then to obtain the mononuclear cells from the blood to characterize mononuclear cells in bulk, and then different immune-based cells for different studies where they were more important to look at. These were subjected to genetic and epigenetic characterization, transcriptional characterization, and immune cell response to vaccine. And I will not be discussing this last aspect of it today. So telomeres are important entities of our chromosomes. Their length dynamics can inform us about biomarkers and determinants of aging and teach us about risk of disease. And also teach us about the interplay between our genetics and the environment. Astronauts in general have shorter telomeres and lower levels of telomerase activity, which is a maintenance enzyme that helps control and regulate the length of telomeres in cells. And our hypothesis is that accelerated telomere shortening and elevated telomerase activity would be associated with the space flight, with the caveat that unfortunately we were not able to detect telomerase activity in the specimens collected because that particular enzyme loses activity when frozen or when during the ambient returns, it lost activity during the duration of the return period. So we were never able to measure that. So we focused, and this was led by Susan Bailey, on measuring telomere lengths as an index of this process. And what we found, and just to remind you again, the green bars are the ground subject, the blue bars are the flight subjects, and this is your pre-flight, in-flight, and post-flight specimens. And what we saw, to our surprise, is that in flight, there was an increase in telomere length, which was unexpected. These results are from peripheral blood mononuclear cells using qPCR, and they were validated using telofish. And as you can see in the ground subject, which is HR, we saw that the telomere lengths over time basically stayed the same, where the red bars are the pre-flight, blue are in-flight, and green are post-flight specimens. But in the flight subject, what we found is that the pre-flight, which is in red, is shifted to larger telomere lengths in blue during flight. And then that pattern is partially reversed, at least in the post-flight specimen where this was measured. This has now been validated in 2 additional astronauts. And so here, the A is the NASA twin study. B and C are 2 additional astronauts who flew for 6 months. And what we see is that there is an increase in telomere lengths in flight in all 3 individuals. This is reversed upon return to Earth. And in this study, as opposed to just having 1 control, there were actually 11 controls, unrelated individuals that served as reference, and there was no significant change in the telomere lengths in those individuals over time. And this particular measurement was now done using multiple different types of approaches, suggesting that this telomere elongation is a biological entity that is seen during space missions. So what else happens at the genetic level during spaceflight? And so what was performed was looking at aberrations in chromosomes, and what we saw is that in the spaceflight subject, there was an increase in the number of chromosomal inversions that could be detected over time. That was not reversible in the post-flight time period. And this, again, has now been validated in those 2 additional astronauts, suggesting that spaceflight of 6 months or longer likely associates with the acquisition of inversions in the genetic material, suggesting that there might be persistent DNA damage likely due to space radiation exposure. So, in addition to radiation exposure, another biological mechanism that could be contributing to this type of phenomenon could be oxidative damage. And so we wanted to look at some of our other data types to see if we could see evidence to suggest that there is oxidative damage or some associated inflammatory markers that are elevated that could also be potentially contributing to this process as well. So looking at urine and plasma specimens in the 1-year mission and in this follow-up study, looking at 2 additional astronauts of 6 months flight duration, we could detect elevated increases in markers of oxidative stress and inflammation that could be detected during flight that then reversed upon return to Earth ultimately, suggesting that during flight, there also might be oxidative stress, and that might also be contributing to some of the genetic changes that we observed during the in-flight status. So this suggests that whether you look at the 1-year mission, the NASA twin study, or the 6-month missions with the 2 additional astronauts, what we see is consistent increase in telomere length, which is represented here and here during the spaceflight time period. And that possibly associates or is due to oxidative stress. Again, that's enhanced during the in-flight time period. And then in addition to that, we also saw these chromosomal inversions in both the 1-year mission and the 2 astronauts of 6-month missions that does not return back to normal, suggesting that these events are truly something that will stay with astronauts long term. So we next wanted to understand whether we could find some evidence of changes in gene expression that could corroborate with some of these findings. So in this plot, what's represented is the changes in gene expression that were observed in the space flight, in the in-flight subject compared to his brother on Earth. And what we found is that there was definitely the acquisition of differentially expressed genes. There were genes that were upregulated. There were genes that were downregulated. Approximately, I think, 6,500 genes in total. And most of these genes, importantly, by 6 months after return to Earth, returned back to the pre-flight status, suggesting that there were definitely flight-associated differentially expressed genes, but that for the most part, these could be reversed within 6 months of returning to Earth. And so what were these genes, and what could they be linked to some of the information that I described previously? So when we performed functional gene set analysis, what we found is that the genes that are subject to differential expression were enriched for genes in immune processes, DNA repair, bone regulation, and hypoxia-hypercapnia responses and processes. And so the changes that we observed in immune cells and DNA repair really further supported evidence to suggest that the processes that I described earlier of enhanced potential inflammatory state and changes to the genetic context could be enhanced during the space flight mission time period. So we wanted to look at some of the additional data types that we had and see if there was additional support for this conclusion. And so the next data type that we assessed were the cytokine data. And this was done using a Luminex assay. And what we found here, and let me just orient you, this side of this heat map is the ground subject. This side of the heat map is the flight subject. And then up here, this pink bar represents the pre-flight, the purple bar represents the in-flight, and the yellow represents the post-flight collections. And what we found when we looked at the cytokine data from this perspective is that there was this pre-flight signature of elevated cytokines in the flight subject compared to his brother, but that signature was enhanced during the in-flight period. And interestingly, in the post-flight period, there was also a more distinct cytokine signature. However, this cytokine signature also was more similar to the ground subject cytokine pattern, suggesting that whatever enhanced cytokine status that was there during flight is largely reversed upon return to Earth. We next looked at some of the metabolites that could be measured in the specimens that we collected. And overall, there were 250 metabolites that were different between the flight subject and the ground subject. However, interestingly, there was an increase in complex lipids in the flight subject. And these complex lipid types that we detected have also been suggested to be associated with increased inflammatory states. So we now had several lines of evidence suggesting that an enhanced inflammatory state is associated with the in-flight time period. And one of the follow-up studies has looked at a variety of data from additional models that have been flown in space. And what's been reported in cell lines and also in vivo models, animal models on the space station, is that there's aberrant expression of immune responses in these models as well. suggesting that this is a phenomenon that is happening biologically across all types of biological systems that have been interrogated. And finally, this heightened inflammatory state has been further assessed in another 59 astronauts. And what was found is that when you compare the in-flight specimens, so the FD specimens, to pre-flight and post-flight, looking at different markers of inflammation, you again see these enhanced inflammatory states during flight time. And one of the other interesting things that we were able to do in the twin study is to actually look at specimens collected immediately and right after return to Earth. And when you do that, we actually found a very return-specific and limited increase in certain cytokines, suggesting that the actual return to Earth, and a couple of days after that, I think we measured it up until day 5 after return, has a very distinct inflammatory response at least in this one subject that was flown. So we further wanted to understand some of the more biochemical aspects of human biology in space, in the spaceflight of the NASA twin study. And to do that, we assessed metabolites and other physiological adaptations during the NASA twin study, and highlighted here are the specimens that were collected for this purpose. And these specimens were subjected to a variety of assays. And I'll just be presenting a small subset of the findings that we had from that particular study section. And this was led by Dr. Brenda Reyna. So one of the things that was pretty obvious is that there was elevated levels of lactic acid in the flight subject during the flight period, and that there was really no change in that essentially in the ground subject. And in addition to this elevated lactic acid level, there was also a decrease in the pyruvic acid level, suggesting there is a disbalance between aerobic and anaerobic metabolism in the flight subject. And potential reasons for that could be weight loss that occurs during flight, the increased exercise regimen that astronauts comply with during flight to maintain their muscle mass, muscle catabolism, or another potential explanation for this would be loss of mitochondrial function. So to assess this last potential, Dr. Reyna utilized a seahorse assay, and I don't have time to go through all the aspects of that assay, but what it does is it measures mitochondrial respiration and as an index of mitochondrial function. And what can happen is if you have a solution or some sort of chemical that can inhibit mitochondrial function, what you see is you'll see changes in the respiratory capacity of the mitochondria in cells when they're exposed to these chemicals. So the question that we wanted to answer was, were there changes in circulating factors in the flight subject that could alter mitochondrial function? And what we saw is in the ground subject, when we exposed the experimental cells to plasma, we saw no change in mitochondrial function over time in pre-flight, in-flight, or post-flight time periods. However, when we took plasma from the twin flight subject, what we found is that over time, there was mitochondrial dysfunction that could be detected using this assay, suggesting that there might be plasma factors circulating that could be responsible for this. And furthermore, what we saw from the sequencing data was that there was elevated levels of circulating cell-free mitochondrial DNA, suggesting that there might be, in addition to mitochondrial dysfunction, there might be mitochondrial damage that could be happening during spaceflight as well. And we don't think that this was kind of a general phenomenon affecting the genome in general, because when we looked at chromosome 21, we did not see elevations or changes in chromosome 21 during the same time period, looking at cell-free DNA. It seemed to be very specific to the mitochondria. So some of the follow-up publications have started to look further into this. And interestingly, when you look at the cell line models that I mentioned before, and you look at the differentially expressed genes that are acquired in the flight specimens compared to the ground specimens, some of the genes that are commonly changed in this situation have to do with mitochondrial function, both ATP synthesis and the electron transport system, as well as genes that are functional in oxidative phosphorylation or the oxidative stress management in cells. And specifically, if you look at the gene expression data from the twin study again, what you find is that in addition to this increase in cell-free mitochondrial DNA, in addition to this dysregulation or malfunction of mitochondria in general when exposed to plasma from the flight time period, you also have increased expression of some of the mitochondrial genes during the flight time period, which are highlighted here in the yellow boxes down here, compared to the black boxes in the pre-flight period. So altogether, what we think some of these things suggest is that there are flight-limited lengthening of telomeres, which appears to be reversible during flight, that space-associated transcriptional reprogramming is largely reversible upon return to Earth. These are all good signs for people who might be traveling in space. And that there are evidence to suggest aberrant mitochondrial function and elevated inflammatory responses and states during the space mission time period. Now, overall, in the twin study, we found many changes that occurred with flight, but important and reassuring for astronauts and us in general is that most of these changes that we detected were within normal limits of variation, and most of them returned to the pre-flight states upon return to Earth. The findings from the NASA twin study have really been hypothesis generating and estimates of potential risk factors for long-term missions. But reassuring is that some of the follow-up studies have been confirming parts of the information that we proposed and identified. And importantly, it was the beginning of the genomics era for space travel, and there have been many, many more studies that have been published that are now generating these type of data from these specimens. From a biological and physiological perspective, the data could mean that there's uncoupling between nuclear and mitochondrial genomes, meaning the mitochondria is damaged and the nucleus is relatively protected other than those inversions that we identified, that there's systemic changes in gene expression that can be linked to energy production and inflammatory states. And this could suggest that there's mitochondrial dysfunction in the sense of misassembly of the respiratory chain and decreased oxidative metabolism that could be contributing to reactive oxygen species, that could be contributing to the damage that we observed. And a shift from glycolysis to oxphos respiration and production of aberrant long-chain fatty acids, which could also be contributing to inflammatory states during travel. together, some of this information could be suggestive that during spaceflight, there is a transient premature aging physiology. And, the question is whether we can come up with ways to change that and prevent that from happening, or at least help facilitate the complete reversal of all of these changes upon return to Earth. Now, I would be remiss without highlighting some of the limitations of the study. It was only one twin. There were no other astronauts tested on the same flight. And clearly between the flight subject and the ground subject, there were confounders such as differences in isolation, food, and activity that occurred. The clinical significance of the findings is unclear because, first of all, the findings could be biomarkers only, may be indications of health, may be useful for interventions, and none of them were done using kind of laboratory based from a clinical perspective results. Finally, it may not be representative of long-term space travel effects since the ISS is still under the protection of Earth's magnetosphere. However, if some of this information will be useful in the future, there needs to be a way to detect some of this data live on the International Space Station. And there have been reports of now sequencing being able to be done on the International Space Station, which really opens the door for utilizing this type of data on the International Space Station in the future for monitoring astronaut health. And so I think, overall, what is the goal of all of this is to help understand space risks and to understand how they change depending on the type of mission that is pursued and the types of risks that people might be subject to towards longer term missions that would start to mimic missions to Mars. But through experimentation and discovery, both on Earth and on the International Space Station, the hope is that we will identify countermeasures that can prevent some of these risks from becoming real health risks for humans in space. And I think I agree with this review article that suggests that we can study these both on Earth and in space, and hopefully do experimental setups and therapeutic intervention approaches that could start to answer the question of whether some of these biological changes that I described and many others that have been reported could be prevented or at least controlled to the point where astronaut health could be protected. So I wanted to thank you for your attention. As mentioned at the beginning, I described a very small part of the NASA Twin Study report. There are many, many papers that have been published since, and I encourage you to explore those if you are interested in this topic more. And again, I wanted to thank all those who were involved and acknowledge the labs that contributed to the data that I discussed today, and the NASA PIs without whom this would have not been possible. And down here, I placed the website for the actual twin study itself for those who might be interested in learning more about it. So thank you for your attention, and I am more than happy to take any questions.
Thank you very much. That was truly inspiring, especially since all of the kind of pop science articles that I've seen, the headlines talk about the aging, they talk about telomere elongation, but not everyone really highlights the mitochondrial dysfunction, which is truly fascinating. I love this. So really, thank you very much for that presentation. And we do have a lot of questions. Probably we'll not be able to go through all of them, but let's try. Okay, so first question, which of the analyzed parameters do you expect to be even more altered after leaving the protective magnetosphere?
Now, if I knew the answer to that, I'd be a rich person, okay? I think it's really hard to predict, but I think the most important parameter that we really couldn't control for in the one of the important parameters that we couldn't control for in the NASA twin study was the radiation exposure, right? Once you're out there, the radiation exposure is at a completely different level. And so I think anything that can be affected by radiation exposure is what's going to be the most different compared to what we observed. And that will range many things, right? Genetic aberrations, damage to tissues. We didn't really measure that, but just like radiation going through tissues of the body and how that will impact like the extracellular matrix and all these other things that can happen. There's also from medicine, of course, we know that radiation can impact vasculature and heart health and things of that nature. And so there's really a variety of things that I think can be further impacted, but I think the radiation exposure is probably the one aspect that we really don't understand fully. And I think that's going to be one of the most damaging aspects of things once you leave the magnetosphere.
That would be truly something to have little PET scanners there on the ISS.
Yeah. Maybe someday, who knows?
Another question. Thank you very much for your lecture, Francine Garrett-Bakelman. Congratulations. From the samples analyzed, is it expected that there is an adaptation of telomerase activity, immune response, and ROS, reactive oxygen species, after a long exposure of astronauts in space?
Yeah, so that would be the prediction. But as I mentioned, we could not measure the telomerase activity. So we don't have direct evidence to answer that question.
Yeah.
Hopefully as the ability to process specimens gets more sophisticated on the space station, we'll be able to start to answer some of those questions. That's an excellent question.
Yeah, that's the hope for the future. Okay, so another one. Do we know already if the ISS-flown twin exhibited accelerated aging from the returning to Earth compared to his brother? Excellent question. I love this.
Yes, I ditto on that. Really good question. So the twin study, the study itself was 2 years, and it started like half a year before the flight, the 1 year of the flight, half a year after the flight, and then it stopped. So I don't have the answer to that. And I don't have a crystal ball to answer that question either. So I don't know.
There were no follow-up studies to the twins?
So the twin study was a 2-year research project.
And that's it. I understand. Okay.
Yeah.
Another question. So you discussed some aspects of human physiology in space, but I'm curious how the endolymph of our inner ear is affected as it alters the perception of balance. Have any endolymph samples been taken from astronauts after their missions in space? Thank you very much in advance.
Yeah. So the answer to that is I don't know, but I don't think so. You should feel free to reach out to NASA researchers who look at this. I'm sure they would be happy to answer that question.
Okay. Yeah. Thank you. That's great. So we can all reach out. So another question. This may be too specific a question, but were there elevated cytokines observed for the space flight, the same ones seen from macrophage activation syndrome?
That's a really good question. I don't know that we looked at the data from that perspective, but it would be relatively simple to answer. So I don't know the answer to that.
So we do know which cytokines specifically.
Yeah, if anybody has research like dealing with this, all that information is published in the supplementary tables of our original NASA study. So feel free to look there, and also at the follow-up studies that have looked at cytokine data.
Okay, great. Thank you. So another question, is there any way to say which of the factors, gravity, confinement, or radiation exposure, is contributing the most to the changes in parameters that you detected?
The short answer is no, right? Because everything happened at the same time. And there's no real good model system on Earth that can mimic that environment to perfection.
I guess the pole stations, right? The ones on the poles could be something about the confinement, isolation.
Yeah, the confinement, I think there are situations where you can mimic it to some extent. But it's confinement in the context of being in space, right? With, very different circadian exposures. And I think it's very hard to isolate these parameters and really determine, which are more than others. I suppose, some things you can learn from other aspects of science. Like, we know there's decades of data that talks about what happens to cells and the body after radiation exposure, right? We have unfortunate accidents on Earth where people got high doses of radiation exposure. We have now cancer survivors who've gone through radiation. And so we kind of know what can happen to people. So that might be something that can be applicable, although the types of radiation, of course, that people will be exposed to up there are different to some of these other situations. So I think it's going to be really hard to tease it out. I think the best way to do this is we need more studies of astronauts to see if some of the findings will be consistently detected, which I'm really happy to see some of these follow-up studies reporting that.
So am I. That would be great to see. Okay, I think we have time for one or two more questions. Let's see. Okay, I can see that we're almost reaching the time. So we unfortunately don't have any more time for the questions. All right, thank you, the moderators. Love it. Okay. The closing ceremony and posters will arrive very, very soon. So if I can please ask not to leave, but just stay, because there is an award ceremony and the closing ceremony for the Life in Space. Thank you, Francine. That was really, really inspiring. I'm sure not only for me, but for the other people who are listening. There will be some questions, I think, posted when the video will show up on YouTube, because I'm pretty sure this video will show up on our site. So I guess I can encourage people to ask questions directly to the researchers, to you or other NASA researchers that participated in the study. And if you feel like you have some time and you would like to answer some additional questions, I'm pretty sure there will be present under the YouTube video. So if at some point you would like to go and maybe answer a couple more, that would be great. And again, thank you very much. That was amazing. So thank you, Francine.
Thank you for the invitation. Wonderful conference. Congratulations.
Thank you.